This is the third video for the unit on gene regulation. In this video, we will examine prokaryotic gene regulation with a focus on transcriptional regulation. In prokaryotes, gene regulation is simpler than in eukaryotes because DNA is not packaged into chromosomes, there is no messenger RNA processing, and there is no nuclear membrane separating transcription and translation. This means that in prokaryotes, gene regulation can occur either at the level of transcription or translation, including preventing translation from occurring or using post-translational modifications similar to eukaryotes. The most common gene regulation method in prokaryotes is regulation of transcriptional initiation. Transcriptional regulation can be either positive control or negative control. In positive gene regulation, there is an activator protein that binds to the activator binding site near the gene's promoter. That activator protein recruits RNA polymerase, increasing its ability to bind to the promoter and increasing the rate of transcription. When the activator protein cannot bind to the activator site, then RNA polymerase has a hard time finding the promoter and transcription occurs very rarely. The shape of the activator protein controls if it can bind DNA or not. Oftentimes, there's a small molecule that will bind to the protein, changing its shape, represented by the blue circle here. These small molecules are required to regulate binding of the activator protein. This is an example of an allosteric effect. In negative regulation, the regulatory molecule is a repressor protein, which binds to the repressor binding site on DNA called an operator sequence. Binding at the operator causes the repressor protein to block RNA polymerase from accessing the promoter region. Since RNA polymerase cannot bind, then transcription does not occur. Similar to the activator protein, the repressor protein has two conformations. One is able to bind DNA and one is not, allowing it to regulate the transcription of the gene. We will use the regulation of the lac operon to examine an example of both positive and negative regulation. An operon is a region of DNA that includes the coding sequences for multiple genes. So the genes are transcribed on one piece of messenger RNA called a polycistronic message. You can see here there are two protein coding genes, LAC-Z and LAC-Y, that make up this operon. The proteins encoded by these two genes are necessary for the cell to degrade lactose sugar for energy. LAC-Z encodes beta-galactosidase, which breaks the disaccharide lactose into two monosaccharides, while the LAC-Y encodes lactose permease, which is a transporter protein that allows lactose to enter the cell in high amounts. You will notice this operon has an operator sequence for binding of a repressor protein and a CRP cyclic AMP binding site for the binding of an activator protein. We will first look at the negative regulation of this operon. Negative regulation of this operon involves the LAC-I protein, which is the repressor protein that binds to the operator site to regulate the gene expression. The key to understanding gene regulation is to understand when the gene products are needed in the cell. The cell does not want to waste energy transcribing and translating genes it does not need. Because the lac operon genes are needed to break down lactose, the cell only needs to express them when lactose is present in the environment. If the proteins are produced when no lactose is present, they would be useless. The LAC-I gene, which is not part of the operon itself, is constantly expressed at a low level, so there is always LAC-I protein found in the cell. When there is no lactose present, the LAC-I protein binds to the operator site and prevents transcription of the genes by blocking the binding of RNA polymerase. With no transcription, there is no messenger RNA to be translated, so no proteins are made. When the environment changes and there is now lactose present, a small amount of lactose forms a molecule called allolactose. The allolactose binds to LAC-I, changing its conformation so it can no longer bind to the operator sequence. Since LAC-I is no longer in the way, RNA polymerase is now able to bind to the promoter and transcribe the necessary genes. Allolactose is called an inducer because it induced transcription by preventing the binding of LAC-I. The lac operon has a second level of regulation that exists, which involves an activator protein. So this second level is positive regulation. The activator protein is called CRP, which stands for cyclic AMP receptor protein. This activator protein binds to a small molecule called cyclic AMP, which allows it to then bind to DNA. As an activator protein, when bound to DNA, it is able to recruit RNA polymerase, increasing transcription of the genes. The CRP cyclic AMP complex controls expression based on the presence or absence of glucose. The role of the cyclic AMP CRP complex is to regulate the lac operon when there are other sources of energy available in the environment. The prokaryotic cell would rather use glucose for energy more than any other sugar. This means that if there's glucose in the environment, then the cell wants to slow transcription of the lac operon, even if lactose is present. This will allow it to preferentially use glucose first. When there are high amounts of glucose in the cell, it causes the cell to make low levels of cyclic AMP. Without much cyclic AMP present, the cyclic AMP is unable to bind to CRP, which means CRP is unable to bind to the activator binding site, and RNA polymerase is not recruited. In this example, the environment does contain lactose, which means allolactose has bound to the lac-I repressor protein and caused it to not bind to the DNA. In this scenario, there would be very low levels of transcription. We have removed the repression, but because RNA polymerase is not recruited, only rarely will it find the promoter region and complete transcription. In the absence of glucose, cyclic AMP levels increase to a high level. CRP binds to cyclic AMP, changing its shape and allowing it to bind to the DNA. This recruits RNA polymerase, resulting in very high levels of transcription and lots of final protein produced. In this scenario, there is low glucose and high lactose levels. However, if lactose was not present, the repressor protein would still be bound to the operator and the lactose operon would not be transcribed, even in the presence of the cyclic AMP CRP complex.